The ECLIA is a simple yet efficient method to quantify cellular levels of MeCP2. The ECLIA is fast, more sensitive, and easier to handle compared to other quantification methods such as the Western blot and the ELISA.
To prepare the washing solution, which is 0.05% tween 20 in PBS, add 500 microliters of tween 20 to 1 liter of PBS, and mix vigorously. Prepare a blocking solution of 3% blocker A in PBS and stir gently. Sterilize the blocking solution by filtration. To prepare the assay diluent solution, 1% blocker A in PBS, add 5 milliliters of blocking solution to 10 milliliters of PBS. Next, thaw the monoclonal mouse anti-MeCP2 antibody on ice. Mix 0.67 microliters of the antibody with 4 milliliters of PBS. Then vortex the solution. To solution coat the 96-well high bind plate, carefully dispense 25 microliters of coating solution into the bottom corner of each well, using a multi-channel pipetter. Tap the 96-well plate gently on each side to ensure that the coating solution covers the bottom of each well. Seal the plate with adhesive foil and incubate overnight at 4 degrees Celsius. Retrieve the 96-well plate from the refrigerator and remove the foil. Remove the